Good day to all. We are the Group 3 and we will be discussing the laboratory exercise number 3, Phycology, Morphology of Algae. Introduction. Algae are protists, an incredibly large group of organisms markedly different features. The form and structure of algae sets them apart from plants. Algae thrive in places where few plants would dare to go. Most species of algae are single-celled microorganisms living in aquatic environments. Algae are primary producers on the bottom of the food chain that feed consumers. Algae are often distinguishable by their color. Significance of the study: to provide enough knowledge about algae, to differentiate a great variety of forms, sizes, and colors of algae. Objectives of the study: to know the different form, structure, and function of algae.
Morphological structure of algae. Cyanobacteria. Cyanobacteria are prokaryotic oxygenic phototrophs that contain a green pigment called chlorophyll and a blue photosynthetic pigment called phycobilins. Prokaryotic means they don't have a membrane-bound nucleus, mitochondria, or other type of membrane-bound organelle. A phototroph is an organism that uses energy from the sun to synthesize organic compounds for food. Cyanobacteria structure. A typical cyanobacteria cell consists of an outer cellular covering, a cytoplasm, or nucleic material. The outer cellular covering consists of mucilaginous layer, which protects the cell from environmental factors, a complex, multi-layered cell wall made of polysaccharides and mucopeptides, and an inner living plasma membrane. These are the basics of cyanobacteria structure. The cytoplasm has pigmented lamellae around its periphery derived from plasma membrane. The nucleoplasm, where the DNA is located, is made up of lots of thread-like fibers or filaments and is in the center of the cell. There is no nuclear boundary or nucleus. The nucleoplasmic material, scattered throughout the cell, splits in two during the cell division process. While cyanobacteria cells don't have organelles like mitochondria, chloroplast, and the plasmic reticulum or Golgi apparatus, which are all found in eukaryotic cells. They both have ribosomes. Ribosomes contain RNA and are responsible for protein synthesis. Ribosomes in cyanobacteria cells are approximately 1/3 smaller than ribosomes in eukaryotic cells, but they perform similar functions. Cyanobacteria characteristics. Defining cyanobacteria characteristics are a tolerance of extreme condition and an ability to exist without vitamins. They use phosphorus, iron, and other micronutrients, and ammonia or nitrate as a nitrogen supply. Some types of cyanobacteria are filamentous and don't need sunlight. Instead, they grow in the dark, relying on sugar from glucose or sucrose as a carbon and energy source. XANTHOPHYTA. XANTHOPHYTA are yellow-green algae that live in fresh water. They may be unicellular in morphology or colonial algae, bunched together. Color is derived from green, yellow, and orange pigments involved in photosynthesis. Flagella make this type of algae multi in water. Thallus structure of Vaucheria, genus of XANTHOPHYTA. The thallus is made of long, cylindrical, well-branched filaments. The filament is aseptate, coenocytic structure. The thallus is attached to substratum by means of branch rhizoids, or branch holdfasts called the haptera. The thallus of B. myandensis is differentiated in subterranean branch rhizodial system and an erect aerial system. The filaments are rough, interwoven, and appear as dark green felt-like structure. The thallus structure is differentiated in cell wall and protoplasm. The cell wall of thallus is thin, weak, non-elastic. The cell wall is made of two layers. The outer layer is peptic, and the inner layer is cellulosic. Inner to the cell wall, there is a thick layer of protoplasm. A very large central valve filled with the cell sap runs from one end of the filament to another, forming a continuous canal or siphon. In the peripheral part of protoplasm are present a large number of small oval or dish-shaped chloroplasts which lack pyrenoids. The chromatophores of Vaucheria contains pigments, chlorophyll A, chlorophyll E, carotenoids, and unknown xanthophyll. The pigments in Vaucheria are like those of Xanthophyceae as chlorophyll B. The characteristic pigment of Chlorophyceae is absent. Many small nuclei lie in the cytoplasm inner to the layer of chloroplasts. The arrangement of nuclei with respect to chloroplasts is reversed at the time of zoospore formation. The cytoplasm also contains other membrane-bound cell organelles such as mitochondria, small vessels, and food is stored in form of oil. The growth of filament is apical. The filament increases in length by apical growth of all the branches. Characteristics of Xanthophyta. A majority of yellow-green algae are freshwater, but some of the species are aerial growing on tree trunks, on damp walls, or soil on drying mud. The cells in multi-Hynera have two unequal flagella attached to the anterior end. One flagellum is tensile type and the other is whiplash type. The pigments are located in discoid chromatophores and consist of chlorophyll A, chlorophyll E, beta-carotene, and xanthophylls. Chlorophyll B is absent. The dominant pigment is xanthophyll, the adenosantine, which provides the cell its characteristic yellow-green color. The sexual reproduction is rare, and it is generally of isogamous type found among the siphonaceous Hynera like Vaucheria.
So diatoms have two different views. We have the valve view and then the girdle view. The valve view meaning when you look at the diatoms from an upward angle, which is the two structures that I mentioned are viewable from that view. And then the girdle view is when you view the diatom from the side, like a horizontal view. So you have the epivalve and the hypovalve, the epitheca and the hypotheca. So the exoskeleton or the frustule, which is also known as the whole part if you mention the whole thing from the whole side, it's called the frustule. It consists of two halves, which is the epitheca and hypotheca, as I have mentioned. The structure of the diatom is fairly similar to that of a petri dish, with the epitheca overlapping the hypotheca. Each theca comprises of a more or less arced valve. So the fourth illustration that we have is the PHAEOPHYTA, which is better known as brown algae. So the structures of PHAEOPHYTA are actually easily observable by the human eye, apart from the things like the bladder and the stipe. But overall, so the brown algae is like the brown seaweeds that you can encounter in the water. So you can see the blades as the leaves of these seaweeds and the stipe as more of the stem or the one that provides structural support for the brown algae itself. And then the last illustration that we have is the RHODOPHYTA, which is better known as the red algae. So as you can see, the structure is very comparable to the brown algae, such as that it is both in the form of seaweeds, as I took this form of red algae for simplicity, as there are so many. So the first distinct features of the seaweed red algae is the blades and the stipes, such as the one in the brown algae seaweeds. So the blade are the leaves, same as the leaves in the brown seaweed, and then the stipe is also the stem. So what makes it different from the brown seaweed is obviously the color, and the red algae are red because of the presence of the pigment of phycoerythrin, and this pigment reflects red light and absorbs blue light, but because blue light penetrates water to a greater depth than light of longer wavelengths, these pigments allow red algae to photosynthesize and live at somewhat greater depths than the other algae. So the red algae can live at deeper sea levels than the brown algae and other seaweeds as well.
Here are the following questions to be answered. Number 1. What is the function of the separation disc and homogeneum in cyanobacteria? Homogeneum is the broken piece of the filament that detaches from parent filament and glides away. The separation disc is the double concave disc of gelatinous material that is formed between cells. It is where the detachment occurs. Number 2. How do you describe the structure of filament and shape of vegetative cells in cyanobacteria? The filament is composed of trichomes and it is surrounded by mucilaginous sheath. It may be branched or unbranched. The shape of vegetative cells could be multicellular or unicellular. It could be a form of colonies and different shapes. Number 3. What is the function of the heterocyst and akinete in cyanobacteria? Some of the filaments of cyanobacteria have special cells, and they are called the heterocysts. It is a barrel-shaped cell that is enlarged. There are different views about its nature. One example is that it has a role in nitrogen fixation. Akinete is a thick-walled reproductive cell that is also enlarged. Its cell wall stores food reserves and enlarans. Number 4. Describe a vegetative branch of yellow-green algae. There are at least 600 species of yellow-green algae. Most of them are found in fresh water. There are different sizes and some of them are single-celled organisms. There are small filamentous forms and simple colonies formed. The XANTHOPHYTA has a cell wall that is composed of cellulose and hemicellulose. Number 5. Describe asexual and sexual reproduction in yellow-green algae. Asexual reproduction, it involves the fragmentation of filaments and spores formation. It may be flagellated or non-flagellated. The sexual reproduction, there are two genera. The Botridium, wherein the sex cells are isogamous, and the Vaucheria, where it is oogamous.
Are there differences between freshwater and marine species? The main difference between freshwater and marine life is the habitat they come from in the wild. Freshwater fish live in streams, rivers, and lakes that have salinity of less than 0.05%. Marine life refers to fish living in oceans and seas. What is frustule in diatoms? A frustule is the hard and porous cell wall or external layer of diatoms. The frustule is composed almost purely of silica, made from silicic acid, and is coated with a layer of substance, which was referred to in the early literature on diatoms as pectin, a fiber most commonly found in cell walls of plants. What are the functions of puncta in the frustule in diatoms? The frustule also contains many pores and slits that provide diatoms access to the external environment for processes such as waste removal and mucilage secretion. What is the substance that is deposited in the cell walls of diatoms? The hallmark of diatoms is their ability to produce cell wall coverage made of silicon dioxide. Silica formation is an intracellular process regulated by the cell cycle. Silicic acid molecules, the bioavailable source of silica in the aquatic environment, are taken up by the cell and then converted into enigmatic process to the convoluted shape of the mature cell wall. Silica deposition occurs inside the specialized organelle silica deposition vesicles and is controlled by the orchestrated activity of macromolecules. So how could you distinguish a plurilocular and unilocular zoosporangium in brown algae? What are produced upon the germination of the diploid and the haploid zoospores? Plurilocular sporangia, a large number of cuboidal cells. Each cell develops into zoospores, develops into sporophyte, more common at warmer temperatures. Unilocular sporangia, one enlarged cell undergoes meiosis, then several mitoses occur to 32 or 64 zoospores, more common at lower temperatures. The small diploid cells undergo meiosis, and among pinnate diatoms, the resulting haploid gametes fuse into a zygote, which grows quite large and forms a special kind of cell called an oxospores.
What is isomorphic alternation of generation?
Isomorphic alternation of generation is a sort of life cycle found in terrestrial plants, and a few green growth in which consequent generation of individuals alternate between haploid and diploid living beings. This generation interchange between the sporophyte able of making spores and the gametophytes able of making gametes. So next, we're going to differentiate the gametophytic and the sporophytic stage in brown algae. So when you say sporophyte, it is carried away from the parent plant by water currents and their own flagella. This is where plants produce diploid which will develop and turn into gametophytes. So gametophytes will differentiate male antheridia from the sperm cells and will originate female ooginum from which the egg cells will originate because it uses mitosis to make haploid gametes. Okay, next. Describe the thallus of red algae. So the thallus are filaments or plates of cells that range in size from a unicellular structure to a complex tree-like form. It can be either thread-like, like in a porphyra, or blade shape. Red algae possess pigments of the picoerythrin and picocyanin group, which involve in photosynthesis.
What is a carposporangium? Explain the function and the role of pericarp and trichogyne in the formation of carpospores in red algae. Carposporangium is one of the sporangia forming the cystocarp in the red algae and containing carpospores. Before fertilization, the first cell of carpogonal branch, known as the basal cell, produces several cells, which eventually form the pericarp. The developing carposporophyte is surrounded by extensive mucilage and elongate pericarp cells, and fuse with the cells of the other sterile branches. Pericarp develops to form a thick protective tissue that surrounds the carposporophyte. Trichogyne is like a hair-like structure which helps in receiving sperm before fertilization occurs, and once it has been fertilized, the cell wall at its base progressively thickens, separating it from the rest of the carpogonium at its base, and after it disappears. What are tetraspores in red algae? Are they haploid or diploid? Tetraspores are found in red algae, which are produced during the tetrasporophytic phase on the algae's life cycle, that also bears tetrasporangia. This phase is also similar in terms of morphology in the gametophyte phase. After meiosis, it produces 4 haploid tetraspores, which will germinate into male or a female gametophyte, then until it matures and fertilization occurs, and produces carposporophyte, and then it produces diploid carpospores that develop into tetrasporophytes. Briefly explain the asexual and sexual mode of reproduction in red algae. Algae is asexually reproduced by ordinary cell division or by fragmentation. Red algae produces monospores in 3 types: walled, non-flagellate, and spherical cells, which are carried by water currents and upon germination produces a new organism. Algae sexually reproduced by meiosis and regulated by environmental events, depending on the environment's temperature, salinity, and inorganic nutrients. In result, algae produces diploid sporophytes and acquires spores within the structure.
So, the experiment for morphology of algae was done by identifying and labeling the parts of its structure of CYANOBACTERIA, XANTHOPHYTA, BACILLARIOPHYTA, PHAEOPHYTA, and RHODOPHYTA in the illustration. In the questions above, the function of separation disc, homogonium, heterocysts, and akinites to cyanobacteria was further discussed. The asexual and sexual reproduction in yellow-green algae was differentiated. The freshwater and marine species was reviewed and were compared, resulting to its distinction based on their habitats. On the midway part of the questions, the diatoms have been researched as well, while on the latter part, isomorphic alternation of generation was investigated and the gametophytic and sporophytic stage in brown algae were differentiated. The form and structure of algae sets them apart from plants due to this experiment. We were familiarized with its structure and able to find and identify the difference of the algae that were used based on their parts.
And thus our report for today ends here. Thank you for listening.